Taste sensation is a fundamental process for us to perceive the food we eat. The taste bud present in our tongue enabled us to experience different tastes, including salty, sweet, umami, bitter, and sour. When the taste molecule binds to the apical part of the taste cell, it causes the activation of intracellular signaling, which eventually transmits the signal to our brain. TRPM5 is a gene critical for the activation of sweet, umami, and bitter sensing cells. In the taste cell, the taste receptor protein can directly recognize various taste molecules and becomes active. This triggers the calcium ions to be released from the endoplasmic reticulum. An increased level of intracellular calcium opens the TRPM5 channel and causes an influx of sodium that depolarizes the cell membrane. Subsequently, the CalHM1 channel opens and releases ATP molecules for downstream signal transduction. Although the activation of TRPM5 is known to be a critical step for taste signaling, the mechanism by which calcium activates the TRPM5 channel is unclear. To understand the calcium-dependent gating mechanisms, we determined high-resolution cryo-EM structures of TRPM5 channel. TRPM5 is a tetramer that contains a pore-forming transmembrane domain and a large intracellular domain. In the presence of calcium, we identified two distinct calcium binding sites for each subunit. We also captured a structure of TRPM5 in complex with its inhibitor called NDNA. In the apo state, both transmembrane and intracellular calcium binding sites are unoccupied, and the channel is not conductive to extracellular sodium ions. Calcium binding to both sides initiates a conformational change such that TRPM5 now becomes open. The NDNA molecule binds to the transmembrane domain of the protein and inhibits the ion flow across the channel. Our study provides a detailed mechanistic understanding of TRPM5 channel function and a foundation for designing TRPM5 specific drugs.